and interesting to see the movement of these very rare diseases get together to form an organisation that essentially supports a very broad spectrum of different diseases, each characterised by the fact that they are rare. None of the diseases that are in the rare category are possibly as rare as some of the diseases that our speaker today works on. Some of the immune diseases are incredibly rare, and some of them actually have perhaps only one family with particular diseases. So it gives me great pleasure to introduce to you today a very long-standing, highly productive researcher at John Curtin School, Professor Matt Cook. So Matt is one of our highly talented individuals. He works in the Immunology group. This is a group that's got lots of grants, has actually made a large number of discoveries, and works on immunodeficiencies and autoimmune diseases. So Matt's interest is in looking at some of the diseases that are caused by mutation genes involved in autoimmunity, and we're going to hear about that from him today. Matt did his PhD at the University of Sydney, trained as a doctor in the University of Sydney, spent a couple of years after getting his fellowships in sunny Birmingham, and then has come back to, or has arrived in Canberra, where he's actually head of the Immunology group at the Canberra Hospital. But also recently he's taken up a very challenging job, and that is he is now Professor of Medicine at the Canberra Hospital and ANU. It's a joint appointment between Canberra Hospital and ANU. So we're looking forward to seeing some interesting contributions from him over that. So Matt, thanks a lot. Looking forward to your talk.
Thanks very much, Simon, for that introduction and for the invitation to give this talk today, which I was delighted to accept. I think that, as Simon has already mentioned, the move towards greater recognition of patients with rare diseases is very welcome. And it leads to the topic of my discussion today, which is a little bit to do with the journey that patients with rare diseases have to endure. And then I'm going to follow on from that with some discussion about why rare diseases are rare, and then what we stand to learn from understanding the mechanisms of rare diseases. Now, here is a list of diseases that cause considerable morbidity, and most people in the room will be familiar with the list of diseases here, and that's because they're not rare diseases, these are diseases, and this is in contrast to what I'll be discussing today. But you can see that overall, collectively, the percent of disability that's accounted for by these diseases does still not amount to a huge proportion of overall human morbidity. Now, rare diseases are defined arbitrarily according to their prevalence, and there are different definitions that have been arrived at by different groups. But usually it's thought that these are diseases where the prevalence is less than 1 in 2,000. And in the United States, this means that there are conditions where the less than 1 in 2,000, and in the United States they also consider a definition fewer than 200,000 people are affected by any given condition. And as Simon mentioned, collectively, rare diseases are not really rare because there are so many diseases that meet these criteria, and so if we add them all together, they account for a very substantial overall burden of human disease. And as I'll go into, they also often begin early in life, and therefore They're chronically debilitating disease. Often there is no cure. And this adds to the burden, certainly, for patients who suffer with these diseases. Now, they're disproportionately represented in severe childhood diseases, so 35% of the deaths in children under the age of 1 year are accounted for by these diseases. Now, some diseases are even so uncommon or poorly understood that they can never be diagnosed. So this is an extreme example of rare diseases where we can't even make a diagnosis at all to arrive at treatment. And patients have to endure ongoing uncertainty about the nature of their condition, what investigations might actually lead them to an answer. This is an ongoing problem. But even in patients who have a recognised condition, the problems that they confront are substantial, and they're really quite different to many of the problems that are confronted by patients with more common conditions. And so here is just some of those. So a delay in diagnosis is something that's reported very commonly. It stands to reason if a patient presents with symptoms which are not easily attributable to a known disease, then it takes some time to find a doctor who's able to understand the nature of those symptoms, arrive at a set of tests that will lead to a diagnosis. And unfortunately, we know that this also often affects the patient outcome, that the delay in diagnosis can result in the development of complications while they're waiting for that diagnosis to be made. There are obstacles to accessing adequate medical care. There are difficulties obtaining affordable treatments. We have a very good healthcare system in this country, but it certainly is oriented towards common diseases, and many of the treatments, when they are available for patients with rare diseases, are difficult to access. And all of this contributes to substantial financial and emotional stress for these patients. Because these are mostly diseases that begin in childhood, at the time that they finish with their paediatric care, they need to make a transition to the adult hospital, and often this is another period of stress because they need to find a doctor who understands their condition, and often in the adult hospital this is to achieve. Now, there are some unique problems for patients with rare diseases in Australia. Our demographics contribute to this. So because we have a relatively small population in a large country, patients have access to different types of medical care depending on where in the country they live. And certainly there are many excellent centres that deal with patients with different types of rare diseases. But of course there's usually only one or two of these, and they're not necessarily just down the road from where the patient lives. And overall, because of the spectrum, the extensive spectrum of rare diseases, we don't always have experts available. Now, when it comes to diagnosis, as I'll come to, often genetic testing is essential, and again, there's inconsistent access to genetic testing in this country. And at the moment, very limited access to what's called genomic testing, where we look not just at individual genes, but variation in all of the genes. And again, I'll talk about this a little bit later. So overall, there's a lack of awareness in the medical profession about all of these conditions. That's understandable. There's a lack of awareness in the general population as well, and this is often frustrating for patients who are dealing with their symptoms. When they talk to their friends about the symptoms that they're suffering with, as we all often do when we're unwell, they also have to often give them a little bit of a lecture on the nature of their condition as well, and so this adds to their overall burden. At the moment, we really don't have a cohesive national approach. In the United States, for example, there are a number of departments and organisations which have been established to try and deal with this problem. And we don't have the equivalent agencies yet in Australia. Now, today's talk here and the other events which are taking place around the country have been supported by Rare Voice Australia, which is an important organisation which is advocating for patients with all sorts of rare diseases and provides support and it is doing a lot to try and rectify this deficiency of agencies that we have for patients with rare diseases. There are support networks for individual diseases, and I must say that in my experience, the advent of social networks has facilitated this enormously. So now patients are able to instigate their own networks much more effectively. I think this is very important. So the title referred to resilience, and that is what I'm referring to, that patients often demonstrate enormous resilience to deal with not only the symptoms and manifestations of the diseases that they endure, but also they have these ongoing problems, the stress often of an uncertain diagnosis, the stress of not necessarily having ready access to diagnostic tests and treatment. Now, in the next part of the talk, I'd like to discuss this question of why some diseases are rare and some diseases are common. But to think about rare diseases, we need to think about diseases in general, and to some extent this seems like it should be a self-evident question, but it is a little bit more complicated than one might think immediately. What is disease? Well, some have defined it as the opposite of health. Not very informative, but this is a definition that's often used. Or it might be health might be a state of complete physical, mental and social well-being, not merely the absence of disease or infirmity. So there's a more comprehensive definition of health. I seldom feel healthy, I must say. Now, we also have this concept of disability, which is any disruption to ongoing personal identity or disruption to the capacity to perform one's normal expected life activities. That's an important concept, but it still doesn't really get us to the nature of disease. So I think a biological concept of disease is a little bit more useful. So a disease is a trait that's more harmful than helpful. We all are a bit different from everyone else, and sometimes those differences are neutral, sometimes they're beneficial, and sometimes they're harmful. Well, a disease is one of those traits that's harmful. And how do we define harm? Well, it means that there is some sort of increased risk of impaired survival or reproduction. And so at the heart of this, there's an incompatibility between an individual and their environment. They're not properly adapted to their environment. And of course, there's biological systems where subject to all sorts of threats within the environment. So there are infectious threats, there are threats from other environmental constituents. So, in essence, what we're talking about here is a disease is where there is some sort of incompatibility between the organism and their environment. Now, how does this come about? Well, here we've got the individual, and they're made up. We're all made up of cells which come together to make organs, and the formation of those cells is specified by our genetic makeup. And as an organism, we live in the environment where we come across all sorts of threats, be they environmental, be they climatic or dietary or potentially infectious threats. And then, of course, we don't usually live in isolation. We have interactions with other members of our species and other organisms as well. So the gene-environment interaction is a complex one. Here we've got a circle indicating us, where we've got our various organ systems, which are specified by our genetic makeup. And some of those organ systems determine how we interact with the environment. So, for example, the immune system, which is specified by a genetic makeup, determines how we deal with microbial threats in the environment. But some of these other environmental threats are dealt with by other physiological systems as well, be it the skin or the endocrine system. And of course, as intelligent beings, we've learned how to manipulate our environment as well. So we're not simply at risk from all of these elements because of problems with physiology. We've learnt to manipulate our environment in certain ways to buffer us from some of these threats. All right, so now if disease is fundamentally arises from this interaction between the individual and the environment, let's think about some concrete examples of how this comes about and why some diseases might be rare and some diseases might be common. Well, there can be exceptional environmental variables. Right? So here are some examples, some substances which occasionally humans come into contact with in the environment. These are rare events, but irrespective of your genetic makeup, usually these are toxic. And so the rarity of any disease that arises from these environmental variables is determined by the rarity of the exposure. It's almost all down to this environmental exposure. We usually refer to these as poisons. But some environmental variables are much more common. Some are even ubiquitous, and yet they're sometimes implicated in disease. And so you will have come across this. You'll have read about or heard about environmental factors that might contribute to cancer or contribute to other sorts of illnesses, and yet they're things that many of us are exposed to all the time. So in this case, we're dealing with environmental variables that are common, but they cause disease in much less than 100% of those that are exposed. Now, here's a particular example. So here we've got some environmental exposures: red meat, dairy products, hamburgers, Diet Coke, fish. Some of these things our mothers probably told us were harmful, but while I wouldn't want them, I think they usually have in mind these sorts of drastic consequences. And so, as a result of exposure to these environmental threats, some individuals, some rare individuals, develop these complications. Now, these individuals have a rare disease called phenylketonuria, PKU. As you can see, it's present in about 1 in 10,000 people, and it results from exposure to dietary proteins that lead to accumulation of amino acids, and normally this particular amino acid is converted to this amino acid in the body, and it makes important chemicals for normal brain function. But in these individuals, there's a defect in the enzyme that's responsible for this conversion, and as a result they accumulate phenylalanine and develop those complications that I mentioned on the previous slide. Now here's another environmental disorder. This one is scurvy. This is a disease that is rare in my experience. I've maybe seen 2 or 3 cases in my clinical career, but it hasn't always been rare. It used to be very common, especially in certain groups, and many of you will be aware that this included sailors. Many of the early expeditions were cut short by morbidity and mortality as a result of scurvy. But now we know that this can be prevented, it can be treated, but it can also be prevented by adequate diet intake of vitamin C. Now, scurvy is the result of the deficiency of ascorbic acid or vitamin C, and this is its normal synthetic pathway. It's a derivative of glucose, and the final step in its synthesis is dependent on, again, an enzyme which is responsible for the chemical reaction that converts this precursor into vitamin C. This enzyme encoded by a gene called GLO. Now, it turns out that none of us have this enzyme, and a number of other species lack it as well. So those shown in grey all lack this GLO gene. So this mutation occurred even before humans evolved, and it's been maintained. Right, so here we've got a couple of different examples of gene-environment interactions. PKU, which is triggered by exposure to common environmental factors, but the genetic basis for this is rare. By contrast, scurvy arises from rare deficiency of vitamin C in the diet, but the genetic factor is, the genetic predisposition is common. In fact, it's ubiquitous. All humans have it. All right, so we'll flesh this out a little bit more, but before going any further, I just need to outline how the genetic makeup works. So we'll just cover this quickly, but you'll be aware that individuals have many different organ systems, and each of those organ systems are made up of different types of cells. And every cell in the body, virtually every cell in the body, carries an individual's genetic makeup. And the genetic makeup is packaged into chromosomes, and what's packaged into chromosomes is the DNA. Now, here's an example of a chromosome. We have 23 pairs of them, but here's one, chromosome 5, and chromosome 5 has about 900 genes on it. And look at this, about 180 million base pairs, these C's, A's, G's or T's. So that's just on one chromosome. So this is the information that's carried in the genetic makeup. We have about 22,000 genes which are distributed across all of the chromosomes, and about 3 billion bases making up the genome. And every time a cell, one of these cells which contains this genetic makeup, divides, it has to replicate all of these genes. So every one of these 3 billion bases needs to be replicated and put in the right slot in the new cell. And this happens in every cell in the body, most cells in the body, through a process of simple replication called mitosis. But it also happens in the cells which we use during reproduction. And there, the process is a little bit more complicated, and it's referred to as meiosis. The genes, the DNA, is made up of this series of bases, as everyone knows, arranged into this double helix. So there's pairing of bases on either side of the helix. But there are only 4 types of bases, and so these 3 billion signals in the DNA are made up of the Gattaca, the G, A, T and C. And DNA encodes proteins, and proteins are made up of amino acids. So we've got 4 different types of bases and about 20 different types of amino acids that are essential. The DNA encodes the makeup of those proteins. But of that 3 billion bases, only about 20% of the DNA sequence goes to directly encoding proteins, The rest is involved in other processes, which I'm not going to go into. So this is a remarkable process which takes place, this replication of all 3 billion bases in every cell that divides, and then also in our germ cells, the sperm and ova. But it's not completely error-proof. There is a small and measurable rate of errors that occur with every replication. And we can now, well, the rate of errors was estimated 70 or 80 years ago, and that estimate turned out to be pretty accurate. And we now know that there are around about 70 to 120 or so new mutations which occur in each generation. And so what happens then? Well, one of the bases in the DNA is substituted for the wrong base, and therefore the code of the protein is altered. Now we've got 4 different types of bases, 20 types of amino acids, and so in order to specify any of the amino acids, it takes 3 different bases. And there's some redundancy here. Obviously you can have 64 different types of 3 base combinations to generate to amino acids, which are the building blocks of the proteins. All right, so the genetic mutations, this change in DNA sequence, arise due to this in replication. And it's variation in sequence, these subtle changes in sequence, which lead to variation between individuals on which selection is made during evolution. But most mutations are serious. And so there is a relation between the number of mutations that an organism can bear, and this is related to the total number of proteins that need to be encoded, and of course the mutation rate. But the upshot from all of this is that changes in DNA sequence are a fact of life, literally. Okay, so I was talking before about phenylketonuria and scurvy as 2 examples of diseases, both rare diseases, but where there were different, the timing of the genetic variation varied a little bit. So in scurvy, the origin of the mutation that leads to the loss of the GLO enzyme occurred even before humans differentiated from, or evolved from other species. For many rare diseases, the mutation that's been transmitted and propagated from generation to generation has arisen much more recently. And to some extent, rare diseases result from mutations which have arisen relatively recently in human history, and common diseases have arisen from mutations that arose a time ago. Now, this is a generalisation. There are some caveats to this, and it also depends on the magnitude of the effect of the mutation, the consequences of that mutation. But it was speculated some time ago that common diseases arose from mutations that are pretty common in the population, that arose from the human population size was quite small. And then as a result of a population explosion, those mutations expanded into many individuals within the population. And we're still seeing the effect of those within the population today. They have a relatively weak effect. There hasn't still been enough time for them to be selected out due to evolution. And so the idea, this hypothesis suggests that the genetic basis of common disease is due to variants which are relatively common within the population. Now, contrast diseases often arise from mutations that arose much more recently, and this is why they are rare. So some families have mutations that only arose 1 or 2 generations earlier, and often these mutations have very, very deleterious effects. This is why the diseases are severe. But they haven't been eliminated through there hasn't been sufficient time for that to occur. Now, in fact, many rare diseases, depending on how we define them, are even rarer, because if we define a rare disease as being due to a single gene defect, and we bring together a group of people who all have that same single gene problem, often the precise nature of the mutation in different individuals is different across that group. Now, we know that some diseases arise from the most recent mutations of all. These are mutations that occurred in the parents, but only in their offspring. And these so-called mutations. So here's an example. Mum and dad are both completely healthy, but their child is affected by some severe disease, and we now know that this mechanism accounts for mental disabilities and some early onset forms of epilepsy. All right, now the final part of the talk returns to this question of Rosetta Stones. How do we learn from rare diseases? What information can be gathered from understanding how rare diseases come about? Well, the important task is to understand how these genetic variants actually lead to disease, and this means understanding the mechanism of disease. Now, I consulted an old textbook which is on my shelf. I actually use this myself, but it's on my shelf, and I looked up the chapter, so this is from 1929, the most authoritative textbook of the time, written by William Osler, and I looked up the chapter on scurvy, and it said the cause is unknown. Nostalgia is sometimes an important element. Now, I know that the effects of nostalgia can be significant, but I wouldn't have put scurvy down to that. And of course, we now know that it isn't due to nostalgia. We've identified the precise molecular pathway that leads to scurvy, and this is important. I also mentioned PKU, and we understand this at the molecular level as well. And this has had important implications because now there's the Guthrie card test, and so all babies are screened for this diagnosis. And by understanding these diseases better, we can also guard against others that might come up with therapies which we know are dubious based on our understanding of these conditions. So how do we make this progress? Well, a significant part of this puzzle has been the resolution of the human genome sequence, which was published in its initial form in the first part of this century. And since then, not only have we identified and characterised the nature of variation in the genomes of many humans, there's also been technical developments which has now made it feasible for everyone to have their genome sequenced for a relatively low cost and in a timely fashion. But in doing this, in sequencing many individuals, we also know that the frequency of variation in the genome between one individual and another is enormous. So the number of single nucleotide variations, the number of bases, those G, C, T or A's that vary from one person to another is very large. And this includes, remarkably, variations in the part of the gene that encodes the protein makeup. Now, this is interesting in its own right, but it also creates problems because if we want to elucidate the mechanism of disease and we're confronted with this avalanche of information, then we need to determine which of these thousands of variants that are present in an individual might actually contribute to the disease. And the complexity doesn't end there because even once we identify in a gene, we need to understand what the protein does. And this is a simplified version of what some proteins do to make a cell work. Here we've got a cell and we've got some pathway. All of the different molecules which are listed here are specified different genes which communicate with one another through these signalling pathways to send signals to the cell to make it function. And so to understand the mechanism of disease once we have the gene, we have to start to dissect some of these pathways. And then, because the body is made up of many different cells which work in different ways, of course, each of the cells in an individual's body contains all the same genetic makeup, but they're switched on and work in different ways, and that's how a heart cell might be different from a nerve cell or a kidney cell. So all of this complexity needs to be resolved. Now, if we think about a disease, and if we think here, each of these nodes might indicate some significant step along the way in such a signalling pathway, as I showed you in the previous diagram, ultimately leading to some change which we might consider to be a diagnosis. And the point of this diagram is to show you that there can be many different paths to one particular outcome along the way. So you might consider this to be a test that a doctor can do on you, which is thought to be crucial in making a diagnosis. It might be a deficiency of one particular cell type in the body. But that can be caused by things going wrong across many different pathways. Now, for most of the past century, the way we've dealt with that when it comes to treatment is to do something like this, where we don't really need to know precisely which pathway has been affected. We can apply a treatment that can disrupt many different pathways, including the one which is causal. But of course, this leads to side effects as well. All right, so if we go through this path, then we can start to resolve disease in a much more precise way. And the reason that understanding rare diseases is crucial and can be informative is because sometimes it not only gives us information about the patient who has the rare disease, but sometimes it gives us information about patients who have more common versions of a similar presentation. So here's an example of a rare immune disease where children present with severe forms of eczema. So a very common clinical problem, but these children have an exceptionally severe version of it. And then on top of the eczema, they often have autoimmune disease. So they have early onset type 1 diabetes and inflammation of the gut. So chronic diarrhea, often with gastrointestinal bleeding. Well, following this process that I mentioned before, individuals with this condition were studied, and the genetic basis for this presentation was resolved. A single gene was found to harbour mutations, different mutations in different patients, to account for its presentation. But identifying the gene didn't solve the mechanism of the disease. It didn't help us understand why these individuals presented with these clinical manifestations. Those insights only came once we understood what the protein does, which is to specify the formation of a particular sort of white blood cell. And then once we knew that, much work has been done to understand how those white blood cells regulate the immune response in everyone. And this has been an incredibly productive line of research over the past decade to understand how these particular white blood cells normally regulate immunity. But the breakthrough came from studying these rare individuals who harboured a mutation in this particular gene. And so ultimately what we'd like to do from this sort of research is to resolve causal pathways. so that individuals who present with ostensibly the same clinical manifestations can be investigated, and we can resolve how different causal pathways might lead to those identical clinical manifestations, and then target treat according to those causal pathways. Now, in immunity, we're doing this here at what's called the Centre for Personalised Immunology, where we're studying individuals who have various sorts of immune diseases like the one that I just outlined, and other immune diseases, many of which remain undiagnosed, in an effort to elucidate the genetic basis of their condition, but then to perform this very complicated task of understanding how that mutation leads to changes in their cellular function in order to cause disease. And you can see that this is a circle, because the idea is that once we make those discoveries and we prove the causal pathway, then we can go back to larger groups of individuals who have a similar clinical presentation and see whether that mechanism applies. All right, so what I've explained today is that rare diseases are defined numerically. Patients with these diseases usually a long path towards achieving a diagnosis, and even once the diagnosis is made, obtaining treatment remains a problem. In some cases, and this is an area which is progressing quite quickly, the genetic cause of these diseases has been identified, and this is progressing rapidly because of the technical developments, which particularly in genomics, the ability to sequence all bases within a person's genome. But the overarching challenge that confronts us now is to link this genetic variation to how the cells in the body work, to understand how the disease comes about in that individual and more generally. So thanks very much for your attention.
Okay, thanks very much, Matt. Does anybody have any questions? Feel free to. Perhaps I could roll with the first one. So a lot of these patients that you look at have got mutations in different genes, giving rise to slightly different clinical diseases. In the past, you've had a small armamentarium of drugs that you've been allowed to use. Now, how are you going to actually treat these patients that have all got what we now believe are different diseases, given the fact that your drug armamentarium is not particularly large?
Well, that's true, although certainly, as you know, with diseases of the immune system, there are many drugs which have been invented over the past 20 years which target individual molecules, and people have been studying how the immune system works normally, and have guessed which particular molecules might be ideal targets for treatments, and then they've gone through the laborious task of making the drug, and then the drug's gone into clinical trials, and then the clinical trials haven't identified any benefit, and then the drug's gone onto the shelf, and that's the end of it. Well, we think that part of the reason for some of those failures might be that we haven't been able to identify patients to be subjected to those clinical trials. So there are many drugs which are reborn, if you like, once we are able to properly identify the targets. The other thing is that some of these pathways do have, while they might ramify enormously, sometimes there are some final common pathways which are crucial, and that might be a final common pathway of a particular cellular subset. And so while the genetic abnormalities might vary a lot from person to person, there might still be a target which is a bit more common across individuals. Correct. Yes. So, yes, so as Medawar said, science is the art of the soluble. So we concentrate on the questions that we think we can solve at the moment, and make progress there. And so, yes, we are deliberately concentrating on those conditions where we think that there is a substantial genetic contribution, because we think that, well, there's already empirical evidence that this is a sound strategy, enables us to understand pathways of disease. But you're exactly right, that's signaling this gene-environment interaction, which is going to be substantial. And now, but I think unless we understand the host thoroughly, it will be difficult to understand that gene-environment interaction properly. So, for example, with HLA associations, I think from very recent data, looking at the nature of autoantigens, it may well be that there is not necessarily a huge contribution from exogenous antigens. It might be the nature of the immune system's ability to tolerise itself to self-antigens, which is still fundamental to these conditions. So, look, we have to make progress in understanding gene-environment interactions, but our view is that one way of achieving that is to understand the host response very thoroughly. Yeah, it's a very good point. And again, this is one of these things that, we're necessarily concentrating on genes that we have a large effect, because at the moment we stand a better chance of identifying those. We're not concentrating on those exclusively, and you make a very good point, because we also think that there are likely to be cases where there are interactions between two modified genes, which lead to the outcome. But it's much harder to prove that. Now, fortunately, we also have some technical elements which allow us to model genetic variations that we identify in people with a particular disease, either in cell lines or in mice, and then that gives us a way of exploring gene-gene interactions in an experimental setting.
Okay. Paul.
You again.
You talked about genes and their products, the proteins. You alluded to the fact that there's a lot of junk like DNA as well. How can you take into account the junk in that, the regulatory elements that actually affect the expression of the genes?
Well, again, it's a very good point. So for everyone else to reiterate, most of the progress so far has been made in identifying changes in the genetic code of that 2% that specifies proteins. But as I also alluded to, we're made up of different types of cells, even though every cell has the same genetic content. And so that tells us that genes are switched on and regulated in their expression in different ways, different sorts of cells. So the question is, how do we start to understand how that sort of process might go wrong? Well, again, we're not ignoring that, and there are many other groups with much more expertise in this area than us that are exploring the regulatory components of the genome.
Further questions? Okay, well, just leaves us to thank Matt for a very clear and very useful presentation. So thanks.
Thank you very much.